Hello, my name is Avgi Apostolakou. I am a PhD student at the National Cappadocian University of Athens, and I will be presenting our work on global network alignment for the identification of conserved Alzheimer-related pathways in Homo sapiens and Caenorhabditis elegans. Alzheimer disease is a neurodegenerative disorder primarily affecting the growing population of people over the age of 65. The pathological hallmarks of AD are amyloid plaques of the abnormally folded A-beta peptide, a cleavage product of amyloid precursor protein APP, and neurofibrillary tangles of the microtubule-associated tau protein. Model organisms are often used for the study of AD, such as the nematode Caenorhabditis elegans, whose favorable features include the presence of the proteins APL-1 and PTL-1, orthologs of the human APP and tau proteins, respectively. Since most proteins don't act alone, but rather it's various protein-protein interactions that govern most biological processes, investigating PPIs in the context of human diseases is essential to fully comprehend them. The availability of organism-wide PPI networks in the past few years has made cross-species network comparison possible. A popular method for comparing networks is global network alignment, which aims at mapping the nodes of two or more networks, and thereby determining topologically and functionally similar regions in those networks. In this work, we performed an in silico construction and comparison of Alzheimer-related PPI networks in Homo sapiens and C. elegans. Our main aim was the discovery of common biological pathways that are conserved in both organisms and are potentially implicated in AD. We hope that the study of such pathways will guide experimental studies on the model organism C. elegans and help in the elucidation of the mechanisms involved in this currently incurable disorder. In the course of this work, 3 network datasets were created, each consisting of a Homo sapiens and a C. elegans PPI network. The first human network was extracted from the amyloid interactome, and it included APP, tau, their interaction partners, as well as any interactions between them. For the C. elegans network, the orthologs of the aforementioned human proteins were recovered, and their interactions, as well as those for the following datasets, were collected from STRING, a database of known and predicted PPIs. We used this dataset to evaluate the GNA algorithms selected and tested. Next, the interaction partners of APL-1 and PTL-1, the C. elegans orthologs of APP and tau, were extracted from STRING. Their human orthologs were found and used to build a human network. This approach provided further validation. Lastly, the 100 partners with the best confidence score of APP and tau for the human network and APL-1 and PTL-1 for the C. elegans network were collected from STRING. This represents the least biased dataset since its creation did not rely on ortholog mapping. We focused our efforts into exploring the elements commonly aligned in the latter 2 approaches. The human network extracted from the amyloid interactome and the homologous C. elegans network were used as a gold standard to evaluate the performance of GNA algorithms. Note that the GNA algorithms were incapable of perfectly aligning the networks because they do a one-to-one alignment of nodes, and several human proteins have multiple orthologs in the C. elegans network. When tested, all algorithms fail to align the networks according to the ortholog mapping when using exclusively topological information, and were only successful when combined with biological information, such as sequence similarity. Magna++ gave the best results by correctly aligning 33 out of 51 pairs of proteins according to their orthologs, and was used for all subsequent alignments. Next, the C. elegans APL-1 and PTL-1 network was aligned with the human network of orthologs, resulting in a connected, unified network. Magna++ succeeded in correctly aligning the majority of orthologs, providing further validation. Lastly, in contrast to the aforementioned datasets, the dataset of the top 100 interaction partners did not rely on the mapping of orthologs, a procedure that inevitably introduced bias. It was therefore our primary focus for the transfer of biological information between the two organisms. Alignment of the two networks resulted in a unified network consisting of 74 aligned protein pairs and 152 conserved interactions. The common elements between the unified network of the later datasets were extracted to create the common network that consists of proteins and interactions well conserved across both organisms. This network was extended with AD-related proteins from the top 100 unified network, resulting in a network of 22 aligned protein pairs and 25 conserved interactions. We searched the literature for information and used it to annotate these interactions. Very limited information could be found for C. elegans. Overall, we observed important pathways that were conserved, such as the APP processing pathway and pathways involving post-translational modifications, including the phosphorylation of both tau and the APP protein. In conclusion, most proteins in the human top 100 network, the least biased approach, were found to be associated with AD. Conserved pathways between Homo sapiens and C. elegans were identified, in which APP, tau, and their orthologs participated. In Homo sapiens, these pathways are related to AD pathogenesis, such as APP processing and phosphorylation of APP and tau, while in C. elegans they have not been investigated. The results of this work will help guide experimental studies on AD in the model organism C. elegans, whose findings may provide insight into the prevention and treatment of AD. Please check out our poster or contact us for more information. Thank you.